Sandy. Horizon. I've been looking briefly at studio apartments, and Loki, I can get this one.
Yeah, I was gonna say I thought you could find one. So how far is it?
Not that far. The issue is, okay, I think if I just hold it right here. Yeah. This is for Sandy, who's not currently online. I have 0 watchers. She's not. She said she'd come.
How's it going?
Oh my goodness gracious. Oh, hi. And Rishi. Yeah, there's Rishi. Truly? Yeah. Who's not online?
Are you gonna hold it like that?
I don't see any. I got a tripod from my friend, and I could not. I just couldn't figure it out. I couldn't do it. No, you tried because Talon and I tried. It didn't happen.
She's about to figure out.
Yeah, and you know what? I'll be grateful. I'll be so grateful. No, because I was just like.
No, because the entire top part is missing.
It's like it must be idiot proof, and I'm the idiot.
But not idiot proof if you forget half of it.
Oh, that's actually smart. I'll try it. Thank you.
If you like put it at an angle, maybe I'll angle up.
Yes.
Oh, okay.
Wait, I think that's perfect. Thank you so much.
Oh my gosh. Right at 5:30. Sorry.
You should know about Sandy said, "Is that Crystal I hear?" Sandy, there's also a chat on the YouTube, and you would be the only person in it.
Hi, Sandy.
Say hi to Sandy. Crystal.
Huh?
That was Talon Sandy.
Hi.
Oh.
Hi.
Yeah, there he is. Is this a good view? Hopefully. Yeah, Sandy said hi. Cool. So exciting.
How's it going? It's going. Oh, thank you.
Excited.
Are you
Excited to
taking?
basically finish my
Are you
thesis
taking neuro?
tonight.
Are you neuro right now?
10
Oh,
pages.
you're done.
Beautiful class. Not going.
Oh,
Not
yeah.
getting
How's
published.
that?
I showed my discussion during our dinner
Uh-huh.
today, and Jake was like, "This is really long." I said, "Yeah, but I can sum it up for you in about 3 seconds."
How long was your
Mine
Yeah,
ended up being
I
4.
kind of saw
but
that one coming.
2 of them,
Just
if I had the
knowing
chance, I would have
her,
like that.
right?
Because like,
Yeah,
they're
yeah.
not like
I think mine's gonna be
Actually,
two.
I have experimental neurobio with Boris.
I think it's fine. My goal
Oh,
is to
yeah?
send it to him by
Is it
tonight or
him or
tomorrow
me?
morning. I need it sent. I need it done.
I thought it was me.
Right.
That's pieces. I thought it was me.
I feel like you should replace me on this campus. Huh?
A couple other questions about John Pop.
Like you know what I mean? Well, those are my grad students.
We're not Boris. Mihich is the one that signed the research approval for me.
Yeah. I didn't know they were going.
I mean, I
Boris
would
was
if I
the
was asked.
neuroscience
Huh?
advisor for deans, faculty advisor for deans before we had a PI. Right?
I think this will be a good
Because remember we talked to him before, yeah. And
it's topical.
I need to eat. I've ate 2
but I
matchas,
think it goes.
1 croissant today. Maybe I woke up around like 9. I've eaten stuff.
All I need to know is who's going to be mad at me if I turn it in late.
You
Like,
don't
who
need to think about
exactly?
it. I've been like chewing on here, yeah. I've just been working on this thesis.
Yeah. I mean, that's fair. I feel like I work better when I'm a little hungry. And that way, a little hungry.
IQ dropped his class. But maybe, I feel like for stuff that's not graded, he might be a little bit better.
Okay, the professors are coming in.
You can take a gap.
Yeah, I think more people showed up than RCP, but I think it's because
know yet. There's some people back in Oregon that I could work for, or Washington,
it's okay. But they
or
only
John
had
Mullins
like, I want to say like 100,
looking at
maybe
labs.
150 to 180 RCP for this, and this seats more. Because this room seats like
Yeah,
500.
exactly. I mean, it was amazing. I wasn't in Portland, I was in Eugene, which is just straight green nature.
I'm glad he got to meet with
Just breathtaking, the scale of everything. I went to the coast twice and did big trips. Absolutely.
And Aaliyah's guys really in the room.
They also have, like, if you go work for a science company, they have a lot of companies in the area. So it's a good place to, like, go do a trip. I think there's reasons to hate it. Like, I get it. I was there during the best part of the year, too. Same. No, I was just
debating what to eat. Like, I want a peanut butter and jelly sandwich. I feel like that would be perfect. Are you gonna go to Chipotle with me after this? Or not?
I agree.
I just want to go to Chipotle.
Did you go to Pike Place? I stayed right next to it, actually.
I'm not coming home. I have to go to lab.
Are you serious?
I just have to start some cultures. Well, we're opposite. You do your lab work in the morning. I do it at night.
No, I do it in like from 10 to 3, like a normal person.
I don't do that. I don't like doing that.
So, did you apply
need
to places?
one road trip.
Where?
I need
Oh, awesome. Which is Dallas?
one.
Okay. And you're
Weird
from
thing.
Dallas.
And another weird thing. No.
Yeah.
Okay.
And another weird thing that costs between 80 and 100.
Hello everyone.
Good medical school.
Settle down slowly. First of all, thank you all for coming. And so it's going to be Dr. Martin Chalfie's presentation about the continuing need for useless knowledge. He received his bachelor's at Harvard, and he later on also did his PhD in physiology with Robert Perlman at Harvard, after which he did a postdoc with Sidney Brenner in Cambridge, where he spent a bit of time, after which he immediately became an assistant professor at Columbia, and he's still there. And he has done a lot, obviously, so can't name all of his awards. But of the most prominent, I think he is, of course, a member of the National Academy of Sciences, a member of the National Academy of Medicine. He's a fellow of the American Academy of the Arts and Sciences. He's a foreign member of the Royal Society. He's a recipient of the E.B. Wilson Medal, and of course, he's a recipient of the 2008 Nobel Prize in Chemistry for the discovery and development of the green fluorescent protein. So, without further ado, Martin Chalfie.
Is this on enough so people can hear me? Good. If not, I was just going to scream. That's okay. The title for this talk, The Continuing Need for Useless Knowledge, actually is a takeoff of an article written in 1939 by this man, Abraham Flexner. How many in the audience are pre-med undergraduates? Great. This is the guy to blame. His committee in the early around 1910, came up with all those pre-med requirements, organic chemistry, physics, biology. He's the guy responsible. But in addition to that, he was the first head, and he actually had the idea for it, for the Institute of Advanced Study at Princeton. And he, this is where Einstein was, Gödel, all these, Oppenheimer, all these incredible physicists and mathematicians. And he wrote this article called The Usefulness of Useless Knowledge in 1939 in Harper's Magazine. And in it he says, as you can see on the slide, intellectual life is on the surface a useless form of activity, but it is unexpectedly the source from which undreamed of utility is derived. I'm going to differ with this a bit, but I do think that his whole idea about useless knowledge, knowledge that we don't know where it's going to be applied, what it's going to be leading to, is really an important thing and something we have to continue. So that's sort of the theme of this. And for the next 5 or 10 minutes, I'm going to give you examples of this. The first actually comes from Flexner's article, where he talks in the article, and he asks this question, at 1939, who's the most important scientist in the world? And the industrialist, George Eastman Kodak, he says, oh, that's easy. I know who the most important scientist in 1939 is. It is Marconi. It's Marconi because he invented the radio and wireless. And that's changed our entire world. He's the most important scientist. Also a Nobel Prize winner. And Flexner writes that he basically says to Eastman, you are so full of it. You're completely wrong. This guy is not it. He's inevitable. Because if it wasn't Marconi, someone else would have developed it. It's basically the technological advance. What was really important for that technological advance was the basic information that underlay it. And that's specifically the work of James Clerk Maxwell and Heinrich Hertz, who came up with the electromagnetic basis of electromagnetic waves, which allows radio and wireless and many other things to be understood. And so those are the most important people here in this story, not the person at the end that made some interesting technology. I would say, however, that not only is fundamental research essential for breakthroughs, but in addition, we need the applications as well. We really do need both. It's not an either or. It's that some people can contribute to the new discoveries, others are going to contribute to the new applications. I'm going to say a couple more examples of people saying exactly the same thing. The next one is from Mark Twain, who in a letter to Helen Keller in 1903, so before this article, it says, it takes 1,000 men to invent a telegraph or a steam engine or a phonograph or a telephone or any other important thing. And the last man gets the credit and you forget the others. All of the basic research, all of the other innovations that led to whatever discovery. This was even said earlier than this by Ben Franklin, who in 1783 is in Paris and he's watching one of the Montgolfier brothers' balloons go up in the air, and somebody in the crowd says to him, "It's the usefulness of a hot air balloon." At which point, Franklin looks at him and says, "What good is a newborn baby?" You never can tell. Another example. I have too many examples, but another example. This is an infographic put together by the Quartzoff company. I'm going to show you the whole infographic and you won't be able to see a thing. There it is. But what this is an infographic of, if you look at the very top, it is what do we need to make an iPhone? What is needed for the iPhone? And as you go down, that's the very top. You see the red arrow there. As you go down, you see all the science that is needed. So just one sort of unit down from there, you get this picture of all the science that is needed to make wireless and computers and lenses and cameras and all the other things that we take for granted in the smartphone that we have. And if you keep going down, you look at the basis of these things, and eventually you wind up at the bottom, and down here, and here you have 500 BCE, the idea of describing the structure of language using rewriting the world's first formal system, the discovery of static electricity, the first lens, the discovery of binary, the discovery of zero. All of these things are needed in order to build up on it. So it's not an overnight success. I want to give you one more example of the importance of basic research. And this was put together, it's a short video, it was put together by a group of graduate students and postdocs at UCSF several years ago for a competition that the Federation of American Societies of Experimental Biology had, which is here. It was Stand Up for Science 2014, and this was what they produced, this very short video. These are the people that did it. Let me just run the video and shut up so you can hear it, hopefully.
It's a sunny day in San Francisco. We set up a table by the water and asked people this question. If it's the year 1960 and you had $10 to give to science, would you spend it on A, developing an affordable treatment for diabetes, or B, figuring out how bacteria protect themselves? The majority of people we surveyed wanted to spend their money on a diabetes treatment, but what really happened 50 years ago? The government decided to spend money on figuring out how bacteria protect themselves against viruses. The National Science Foundation, the NSF, and the National Institutes of Health, NIH, are two government agencies that allocate tax dollars to scientists. And they decided to fund groups of researchers who thought that studying how microbes protect themselves was an interesting question. At the time, there was no way of knowing how this basic research could affect human health. They were simply curious about understanding something about bacteria biology. So what did the scientists find? Normally, when a virus enters a bacteria, it adds its own DNA to that of the bacteria. In doing so, the virus takes over the bacteria and uses it to make more viruses. Scientists found that bacteria protect themselves from viruses by selectively cutting the viral DNA. This basic finding led to a scientific revolution. Scientists learned how to cut specific chunks of DNA, and they could use this technique to move genes around. They could even move human DNA into bacteria. Because bacteria are very good at making more of themselves, scientists can use them to make large quantities of human proteins from the human gene that they introduced. So why was this technique useful for modern medicine? Well, one breakthrough was the efficient production of human insulin by introducing the human insulin gene into bacteria. This was a very significant advancement in diabetes treatment. Before using bacteria to produce large quantities of human insulin, insulin was expensive, unsafe, and less effective because it was harvested from fetal cows. In fact, the first person to get an injection of cow insulin suffered from a severe allergic reaction. Because of a long line of federally funded basic research, human insulin is now an affordable treatment for diabetes. By asking how bacteria protect themselves, the scientists discovered that bacteria can cut DNA. This discovery was developed into a basic technique used in thousands of labs around the world to create new knowledge and to ask new questions that have led to all kinds of therapies and technologies, including insulin, DNA fingerprinting and biofuels, the human genome, growth hormones, cancer drugs, stroke medication, vaccines, HIV meds, antibiotics, and many, many more. Given enough time and funding, who knows where it's to come. Many other health benefits and current discoveries would have been nearly impossible without federal funding for all kinds of basic research. Basic research is an investment in our future. It broadens our understanding of the world we live in, and the potential benefits are limitless. Unfortunately, science funding in the US has plateaued, decreasing purchasing power and significantly hurting our competitiveness and innovation. You may not know it, but you and your tax dollars are part of the discovery process. Thanks to you, scientists discovered that bacteria can cut DNA. And thanks to you, insulin, other drugs and diagnostic tools have been developed and are helping patients.
So thank you for supporting basic research. We're the scientists who depend on you, and we are excited for the future that lies ahead.
Pretty amazing video, I have to say. Anyway, if it's important to have these discoveries, how do we make them? I'm going to spend a lot more of the time on maybe approaches to making these discoveries. And I want to first have a distinction between measurements and discoveries. And this actually comes from the physicist Enrico Fermi, who said this wonderful line. He said, if the result of an experiment confirms your hypothesis, then you've made a measurement. If the result is contrary to the hypothesis, then you've made a discovery. So you do the usual things and you find the usual things, good for you. But there are a lot of unusual discoveries to be made. So one of the questions we try to address is how does one make those discoveries? But I want to give some examples from the Nobel Prize of these serendipitous discoveries. It's not so much that it's chance, it's that something happened that was unexpected and they followed up on it. And I think that's the importance. So the first one I want to give is Robert Wilson and Arnold Penzias, who were working at Bell Labs, and Robert Wilson sort of had a failed PhD project. He got his PhD, he was a smart guy, but the project didn't give him what he wanted, and he discovered that the Bell Labs, somebody else previously, had made a 30-foot microwave telescope. And he realized he could use that to answer the question that still dogged him from his PhD thesis. And so he and Arnold Penzias got this 30-foot thing out, cleaned it up, get it all working and everything, and pointed it towards the Milky Way, which gave a very nice, strong microwave signal, which is what they expected. But they had to calibrate the machine, so they pointed away from the Milky Way into empty space, and it doesn't go to zero. They can never get it to go to zero. They first think it's the birds flying around, maybe it's an airplane from the nearby airport, or the power lines that are somewhat near. It's none of those things. They eventually get rid of all the other possibilities and realize that what they're really looking at is something that can't go to zero because it has a measurable quantity. It is the background radiation in the cosmos, which they realized, and other people had talked about, was an evidence of the Big Bang. And so they didn't actually do an experiment. They were just trying to calibrate the machine, and it didn't quite work. Another example is John O'Keefe, who discovered what are called place cells in the brain. And if you read his Nobel Prize lecture, or maybe it's his biography, it's very interesting. He starts it off by basically saying, I was doing some work in looking at the electrophysiology of cells in the thalamus, when one day, by mistake, I put the electrode in the wrong place. And he discovers these cells that eventually gets him the Nobel Prize. But probably my favorite of these discoveries that were made is the co-winner, one of the co-winners was Osamu Shimomura, who discovered green fluorescent protein. He actually discovered more than green fluorescent protein. And I want to urge you, if you can, his daughter and her husband convinced him to write a memoir. It's called Luminous Pursuit. It's really a quite wonderful book, but it talks about the fact that at the age of 16, he's told he has to quit high school and start working in a factory. The reason for that is, well, a war was going on. In fact, the city he lived in was Nagasaki, Japan. And by working in the factory, he actually was saved when the atomic bomb destroyed the city. He writes about walking home the 10 miles from the factory to his home, and by that time his white shirt had turned gray from the radioactive fallout, the ash that had been in the sky. And he credits his grandmother with making him take a bath and probably saved him from radiation sickness. But there's no place to go to college. There's nothing to do. So he goes and he works. He works for a while and he gets to go to the first available university when they rebuild it, and that's the pharmacy school. He said that he wasn't really interested, but it was the only college he could go to. So he goes to pharmacy school, graduates, gets a job as a technician. And in that lab at Nagoya University, he goes and he finds a problem that's an absolutely fascinating biological problem. Many different types of organisms, fireflies, glowworms, fish, different types of organisms, fungi, bacteria, can actually generate light, bioluminescence. So how does this come about? What's the biochemistry behind this? People had known about the biochemistry of fireflies, but it turns out that knowing fireflies gives you no information at all about the others. It's all unique. So the light is being produced in a lot of different ways. And he starts working on a project, and another sort of accidental thing, he actually discovers the right molecule that actually is needed to generate light. And this gets him invited to come to the United States to do more work. And his boss, who's a technician, his boss realizing that his salary will be doubled if he comes to the United States with a PhD. Double, it was $3,000 a year, it could be $6,000 a year if he had a PhD. So he arranged to give him the university to give him his PhD as a going away present. So he does that and he starts to work at Friday Harbor Lab, Washington State, to try to figure out what makes this jellyfish bioluminescent. Why is it producing light? It's known that it produces a beautiful green light, and he starts off grinding up jellyfish. He's a very good biochemist. He does the experiments repeatedly. He starts bribing his kids to find more jellyfish for him. He does all sorts of things, and he goes nowhere. Nothing works at all. He can't get it to work, tries everything he can imagine. One night, the experiment has failed again. He comes, he decides, that's it, I want to go home for dinner. He cleans up after himself. He takes all the samples and he throws them away in the sink, which had some seawater and probably some other stuff in it, and turns off the light, and he's about to leave. It's dark everywhere. Looks back at the sink, and it's glowing bright. That was a surprise. And so he says to himself, I wonder what that was. Seawater. I wonder if seawater has anything I haven't tried in the experiments. He realizes that seawater has calcium ions. That calcium ions might be important for generating the light. Goes back after a couple of days, redoes the experiment, and this time, every time he adds a solution of calcium ions, he gets a spark of light. That was what the missing thing was, and it allows him to purify the protein, which he names after the jellyfish, and that is corn. So corn plus calcium will generate light. So that was the first accidental discovery. But this actually tells you the second accidental problem. It's the wrong color. It's blue light that the damn things are. He adds calcium to all the other samples. Nothing works. The jellyfish gives off green light, not blue. So he has to think about that accidental discovery. By the way, we often talk about the scientific method. You know, you come up with a hypothesis, and then you test the hypothesis and all. But throwing things in the sink around the floor perfectly sometimes work, or I will laugh. Anyway, he thinks about this for a while and realizes maybe there's something that actually converts the blue light to green light. So he takes a handheld lamp, and now he looks at all the samples, and a completely different one. Doesn't need calcium, doesn't need a cord, doesn't need anything. But whenever he shines blue light on it, or ultraviolet light, he gets green light coming back. In 1962, he writes about these discoveries in a paper that probably no one has ever read, but in that paper, footnote number 3 says, the paper's all about a cord, but footnote number 3 says, by the way, there is this other protein, and if you shine blue light on it, you get green. I'm going to call it the green protein. Of course, we call it GFP, green fluorescent protein. And that was the discovery of GFP, and the wrong color. But with GFP, the light from a corn plus calcium will generate green light. I heard about this in 1989, and I'm working on C. elegans, which is a transparent animal, and I wanted to know what genes were expressed. This was the standard scientific talk. I'd say no one had ever wanted to know that question before, but that's a complete lie because people were doing that all the time. People wanted to know where genes were expressed. They make an antibody to look at the protein. Or they do in situ hybridization, or they use a marker like LAC-Z to have some sort of reporter. But here is something that could work in a transparent animal. And in fact, it would be great because all you'd have to do if you had the molecule was send blue light into it and you'd get green light out. You'd be able to see it. Maybe you'd even see it in living animals. I got really excited. I had no idea what the seminar was about. I just started fantasizing about it. And I want to point out one thing. This is probably the only molecule that looks like it should in the world because it's 11 beta strands making a cylinder capped on both the top and bottom, and a single alpha helix going through the middle, and the middle of that alpha helix has a rearrangement, and that's where all the light is from. And so what it actually is exactly what it looks like, a lantern. It's really quite remarkable. I don't know of any other molecule that looks like this. Anyway, let me go on and say a couple of things. Because GFP allows one to look at gene expression. You have a gene, and you can put GFP there, and you can make a transcriptional reporter and see where, when, and how much a gene is active in different cells. And so here's an example of that. Edith Skirkin was the graduate student in my lab who did this. Douglas Crasher was a collaborator that got the first cDNA for GFP. I should say something about my color code. Anyone whose name is in red is someone from my lab. Anyone whose name is in blue is a collaborator. And anyone whose name is in black did something I wish I had done. So here you can see C. elegans with the nerve cells we like in it, with the transcriptional reporter. But you can also put GFP as a translational reporter, and have the protein make GFP, but have the protein made as well. And then they ask, what is its function or where does it go in the cell? And the first person to do that is Tula Hazelrig, who looked in Drosophila and found that these proteins that were labeled with GFP were being transported out into the developing oocyte. Tula is my wife. Let me show you a couple of quick movies made by at the time she was a graduate student in Canada. I've never met her, but she nicely put both of these movies on the website of the American Society of Cell Biology. On the left, we have GFP attached to beta tubulin, so it'll be part of the spindle. Now, in the early development, oh, there we go. You can see the spindles forming. It's obviously very sped up. But the one really remarkable thing about this is what she used as the name of this movie, she calls it In Synchrony. And that really is the remarkable thing here, is everything is happening together. And it immediately invites questions of how the hell does that happen? What are the molecules that allow for this sort of coordination over time? Now, on the right is also GFP, but it's false, the image is falsely colored. So GFP is attached to a small peptide called the nuclear localization peptide that puts it inside the nucleus. But when the nucleus comes apart during cell division or nuclear division, then it's going to go all over the embryo. And so what she's done is falsely colored it. The convention is the more you have, the more you're towards the red part of the rainbow. The less you have, the more you're towards the ultraviolet end of the rainbow. And so let's watch that. Nuclei fall apart, now it's everywhere in the embryo. And the nuclei are going to reform and suck up the GFP. There she calls this after the fading starry night. Looks like that a bit. But what you see here is that it's not synchronous. It's actually going from the lower right to the upper left-hand corner. And you can ask yourself the question, how is that? Why does that happen? What we think, again, we never asked her, is that maybe her, at least from friends who've done similar experiments, they think her thumb was a little heavy on the slide and she was making caused a little bit of damage here. But nonetheless, what a wonderful way of being able to detect whether things are working correctly or not. I want to talk a little bit more about GFP, because why not? Okay, once you have the tool, you can change it. How many of you grew up with the kid's book, If You Give a Mouse a Cookie, right? Yeah, almost everybody, right? And that's a horrible book. You give the mouse a cookie, and then it wants a glass of milk, and then it wants a straw, and then it wants a napkin. It just keeps going until again it wants a cookie. So it's a great metaphor for scientists. Because you give them a perfectly good green fluorescent protein, and the very first thing they say to you is, you got any other colors? And the person who really put that together was Roger Chen, who was the third person to share the Nobel Prize. He mutated GFP to make the first different fluorescent protein, which was the blue fluorescent protein, BFP, and then later was able to make a sort of green-yellow one, which was called YFP. And that was it. It couldn't get any more. People really wanted to get more colors and everything. There are other things that were changed. I won't go into that. And then these two characters, Michael Max, maybe here. Yeah. Oh, and there he is. Yes. You're gonna have to correct me when I get all this wrong. But they realized that corals were fluorescent, that corals had other colors, and that maybe they had DNA with a sequence that was similar to GFP that they could pick out and take a look at. And so they, that's what they did. And they were able to find the first red fluorescent protein, DS red. This went through a number of different changes. Other things were made. I'm abbreviating a lot of this, but this is from Roger's Nobel. He got tired of naming them after letters and wanted to name them after fruits and vegetables and stuff like that, and tomato and grape and cherry and tangerine and all of that, but many different colors. So what happens? Look at this. There's over 1,000 fluorescent proteins listed on fpbase.org, and all sorts of things that people have found in the wild, that people have manipulated to have other properties and so on. So what happens when you get a lot of different colors? Well, one thing is you get definitely sayings that came together and decided to label nerve cells in the mouse brain, and you get brainbow, it's a great name, and it's been used in flies and zebrafish to sort of try to look at the way brains are connected and how cells are developing. Because now you have a way of looking at different things. This is just 4 colors that are mixed and matched randomly so that they have different things. Another experiment that I quite like is by female mammals have 2 X's, but at the 20 cell stage, one of them is turned off in every cell. But which X gets turned off is random, and are there cells that have both on? Well, what these guys did is they put one X expressing RFP, the other one GFP, and what you can see here is really separated. It's quite wonderful. And they looked at many other tissues. Some other things. Okay, I'm going to talk about 2, what I would call inventive uses of GFP. One is by Jeff Waldo, who was at the Alamos Lab. I think he's still there. And Jeff was interested in a really interesting problem. People like to make proteins in E. coli and generate a lot of proteins. But the problem is, if you start making too many of them, they aggregate, they precipitate out. You don't want that. You want them to be soluble. And maybe you could mutate the sequence so that it folded better. And so his idea was quite wonderful. He decided to make a fusion that would encode the protein he was interested in and GFP. But if the protein he was interested in didn't fold correctly, GFP wouldn't fold correctly. And if it didn't fold correctly, it wouldn't be fluorescent. So you could shine all the blue light on it you wanted, you'd get nothing out of it. But if you mutated his protein that didn't fold so it would fold, then not only would that protein fold, but that would allow GFP to fold. And now you can see which colonies, which bacterial colonies had this, simply by shining blue light and looking for the green light. So a wonderful monitor for protein folding. I thought was a wonderfully clever experiment. Another wonderfully clever experiment was done by Lynn Regan, who was the chair of chemistry at Yale. She did a wonderful experiment that at first one would think is not really very good. What she did is she took GFP and cut it in half. If you cut GFP in half, you don't have any fluorescence. But then she did something really interesting. She took onto each half and she added linker groups for leucine zipper domains that would come together. If those 2 domains came together, she found that they would bring GFP together and it would be fluorescent. So this is actually a monitor for protein-protein interactions. You put one part of GFP on one part of GFP on the other, they come together. If they come close enough to each other, you're going to make a fluorescent product. Very inventive, things I never would have imagined. And that's, I think, one of the lessons when new ideas and new techniques are made, is that things come up because other people are much cleverer than you are. So some other interesting uses, there's a group in Japan that made silk moths that would make fluorescent silk and then made clothing out of it. I'm not so sure about this, only because it's so cool, but you have to carry around with ultraviolet light. How interesting your clothes are. So maybe not as good, but quite interesting. There have been people that have made cellular lasers with GFP. Bob Berlage knew that if he took E. coli and put them under GFP under a promoter that was turned on by the presence of the explosive TNT, he would, and he also knew that landmines leak TNT, that if he put that into E. coli, he could actually generate a way of seeing whether a landmine was in the area. And so he put this together. This is from a 3x5 meter plot of land that he had a colleague bury landmines. They had TNT, but they were not live. He was safe, but they were going to leak it, and he sprayed his bacteria, went back with an provide a lamp and was able to show this. This has been done now 3 or 4 times by different groups in different ways. The problem is, it's not, as far as I know, it's not quite ready because you do not want to have a false negative. So the false negative is a disaster. So false positives you don't care about. False negatives here would be very bad. GFP has also gone into the world of art. The Brazilian artist commissioned a French biotech company to make him Alba, the GFP bunny, that he would take to art shows to get people talking about the connections between art and technology and art and science. And finally, somebody gave me a comic to do this. This is maybe the first. If you see the movie Home with Aang, that was done by Aang Lee, it actually starts out with a jellyfish. A hydrocarbon comes in, sucks out the GFP. And it's clear that that's the problem with Bruce Banner. He got injected with GFP and it went to some gene in his skin that gets turned on when he's angry, and that's how we think. I actually know the writer for this play. He's a professor at Columbia, James Shamus, and his daughter and my daughter went to elementary school. After I saw the movie, I went to him and I said, I really like the beginning. That was terrific. How do you know about my work? And he looked at me and he went, I have no idea what you're doing. And I said, oh, that stuff with GFP. So we had a student from MIT that was working on the set, and he convinced us to do that. Fine. Terrific. That was great. Anyway, there are more examples on Mark Zimmer is a Connecticut college. He has a GFP website if you want to look at other examples here. Let me go on from here. So if these discoveries are accidental, how do we get ourselves to make these accidental discoveries? So I think one, and I'm going to talk a little bit more about this, is that I feel that there are questions that are right in front of us that we have become oblivious to, but are actually fascinating questions. I'll give you some examples in a moment. Another thing is I think we need to question our assumptions about what there is. And I think both of these are good approaches. There are other things, let me just talk about the questions right in front of us. My first successful, it's an important word, successful lab experience was working on the major transparent part of the body. So I like to have a little show of hands here. If you know what the major transparent part of the body is, raise your hand. This is what usually happens. It's about 3 to 5%. So I'm going to give everybody else a hint. It's right in front of your eyes. Yeah. Because what it is, this is the cornea. You have been looking through your eyes your entire lives. But the realization that that's a transparent tissue, that the lens is transparent, and asking the question, how does that happen? Is really a wonderful scientific question. When I went and got my first job in the lab, in this lab, I was completely dumbstruck by it. Why didn't I ever think about that before? And there's a lot of other examples, but I'm going to talk about a cousin of mine. Because some of these examples. Ray Goldstein is the professor of physics at Cambridge University. And Ray has, I believe, made a career of asking very interesting physics questions about things that are all around us. One of the really nice things he did was he tried to answer the question, why do ponytails have the shape that they do? What he was really trying to do was try to find out what happens when you have an array of fibers and they're all interacting together. Why do you get the shape like a ponytail? This, by the way, allowed him to win the Nobel Prize in Physics. So, very proud of that. But he's asked other very interesting questions that are all around us. So one of them is, why are icicles shaped the way they do? These are all things he's published on, by the way, not just questions he's asked. Another question, why don't clothes fall apart? Clothes are made up of actually very tiny fibers. So why aren't we all standing or sitting here naked? He's trying to address that. Another thing, more recent, he and his wife asked a question about the emergence of cicadas. Not why they come out every 17 years, but why do they come out in a group? And there's no reason they should come out in a group unless there's some sort of communication. That's what their paper is. All of these are things that are in the world around us. A couple other things, but one that I like, I was a graduate student that was teaching the intro physiology course, and the professor in charge of the class started talking about muscle. There was fast twitch muscle and slow twitch muscle. Fast twitch muscle only worked for a short time to go into oxygen. That slow twitch muscle needed to have an oxygen supply, so it had myoglobin, and by this time everyone in the room was asleep. But it's sort of late October, early November when he's giving the talk, and he stops and he says, and it's because of this that you have dark meat and light meat in the turkey or the chicken. So the idea of being able to, you looked at chicken and turkey all your lives, you know that there's light meat and dark meat. You probably have never asked the question, what is the difference between this? Why is it dark meat? Why is it light meat? Anyway, another question, but I want to give another example, which is this wonderful example from a high school student, Abigail Freed, who I met at the International Science and Engineering Fair several years ago. I was roaming around the session and looking at the various displays, and she was sitting there next to her display, and I stopped and I said, tell me about what you have. And she led off with the most surprising way to start any scientific discussion. Her first sentence was, well, you should know about me that I'm a ballerina. And I'm a very good ballerina, virtually at a professional grade. And ballerinas have a problem. That is that the shoes fall apart after a single performance. So they have to spend $125, maybe more dollars every performance to just get shoes because the cardboard soles lose their flexibility. It's a complete waste. And so she said, and so that was my project. My project was to come up with a material that could be really good for the sole, but would last more than one performance. So she got a carbon composite thing and looked for 1, 3, and 5 ply of it, and found that the 3 ply was the best. And she now had ballet shoes that would work for months and all be the perfect flexibility. I thought this was a great project. Came up with a problem no one else probably would be thinking about, came up with a solution. I thought it was just spectacular. Okay, I'm going to the idea of questioning assumptions. There's a sort of set of puzzles. I liked them as a kid. I still like them. These are called matchstick puzzles, where they give you a set of matchsticks and they say, here's this pattern, now make this pattern. And so to give you a couple of examples, they might say, move 3 matches to get 3 perfect squares. And you sit there and you move them and you yell at yourself or the screen or whatever. And you finally realize that you can move them like this to make the 3 perfect squares. And then once you get sort of confident that you got that done, the next thing is they give you this puzzle, which is a fish. And they say, move 3 matches and make the fish go in the other direction. And a little harder, again, scream and yell, and finally you decide that the way that you're going to do it is that way. You remove 3 matches and you can do it. And now you're pretty confident. But this is where questioning your assumptions really comes in. Because the next problem you might be given is, questioning the assumption is that. Make 4 equilateral triangles from 6 matches. And you move them around and you move them around, and either you quit or you suddenly realize that maybe your assumptions were not right. And that you were thinking only in 2 dimensions. If you don't think in 2 dimensions, you can make the thing as a 3-dimensional model. So the assumption was, no one told you it was 2 dimensions, you just bought it. Because the other puzzles had that. But this actually isn't the only solution. You can do something on the surface, but it requires getting rid of another assumption here. And that assumption is, why are the triangles all the same size? They don't have to be. You could maybe, especially if you have very thin matches, you could make that. And that would give you 4 triangles, 2 of one size and 2 of another. And that's pretty good. And so you sort of say, okay, that's enough. You fooled me enough. And then they give you one more puzzle. And that puzzle is this one. Make these 3 matches into 6. You cannot break a match. Okay? If you think you know what the answer is to this, raise your hand. Yeah.
The Roman numeral 6?
Yes. Why are you assuming that you have to have shapes? It could be the Roman numeral 6. So all of these are the assumptions that we don't even realize we have, but that we have to really question, I think, in all of our science as we think about this. I have a good friend who used to be a colleague. He's now at Berkeley and also in China, Muming Pu, an astonishing neurobiologist. Muming, one day we were all sitting around with Muming, and we said, Muming, you always come up with these fantastic ideas. Where do you get your ideas? Everybody's always searching for more ideas. He said, oh, I do something called the retrospective approach. In the retrospective approach, what he does is he looks for an interesting hypothesis in a textbook. And for him, it was the 2nd edition of the molecular biology of the cell. And he reads it, and once he finds that assumption, he simply says, how do we know that? And he finds usually there was some old experiment that somebody did and then extrapolated enormously to get the hypothesis that they did. And he realizes, well, wait a minute, that was a long time ago when they didn't know how to do this or that or this other thing. I can retest it using more modern methods. If it works, I have confirmatory experiments. I can publish that. If it doesn't work, I've just been able to change how people think about it. So he publishes the correction. And it goes back to the textbook again and finds out what other nonsense we've been believing and trying to get into all of the heads of the undergraduates. I want to end with 3 quotes that I find very nice. Not all related to things, but just I like them. The first is by a different Robert Wilson, Robert R. Wilson. The other one was Robert W. Wilson. Robert Wilson was an incredible guy. He was was a sculptor, an architect. He was the first head of what was called the Fermi Lab in Batavia, which until the CERN Super Collider was the biggest particle accelerator in the world. And in 1969, he was asked to justify Congress actually spending money to make this particle accelerator, which still is producing wonderful data. And he met with a joint committee of Congress, and John Pastore, the senator from Rhode Island, who was in charge of the committee, thinking he was giving him a softball question. easy one to convince everybody else. Remember, 1969, this is in the middle of the war with Vietnam. So he says, Dr. Wilson, can you tell me how many ways this work, this particle accelerator, is going to have national security? How many ways? And Wilson looks at it and says, none. Not the answer he wanted. So he asked it again and then again. Finally he asked it, he said, no, in what respect will this particle accelerator help the national defense? At which point Wilson said the following. It is only to do with the respect with which we regard one another, the dignity of man, our love of culture. It has to do with, are we good painters, good sculptors, great poets? I mean all the things we really venerate in our country and are patriotic about. It has nothing to do directly with defending the country, except to make it worth defending. My first quote. My second quote is from this character, who happens to be my daughter. But it's my daughter in 2004, when she was 12 years old. And that was the time that I was asked, I was elected to the National Academy of Sciences. If you ever have problems falling asleep, try to get a recording of the induction ceremony. Now it's about 120 people, that is about 80 people that get inducted. Everybody in the audience is there for one. They have to sit through the other 79. And what they're sitting through is that one at a time, people get up on the stage, shake hands with the president of the National Academy, have a picture taken with the president of the National Academy, walk across the stage, and there's a book that they're supposed to sign. Told at the beginning, everybody has signed this book. So you're saying to yourself, I'm going to sign a book that Einstein signed. And then they tell you they only started the book in 1970. But as you walk across the stage, somebody has something written up, and they read a sentence about what you got elected. And I was 12 years old. I knew that she was going to be completely bored. So when I finally did my bit and got down, I ran up to her and I said, "This way, I'm really happy that you and Mom are here. I'm very happy that you're here to help me celebrate this. But I just want to apologize to you because this must have been a dreadful thing to have to sit through." And she looked at me, and she has often looked at me like I am the stupidest person on the planet, and said to me, "Dad, don't you understand these people? You don't understand about these people." And I said, "Play along." What do you mean? She'd been listening to all those sentences. And what she got out of all those sentences describing the work that people did was this. She said, they were the first people in the world to see what you were doing. That is one of the joys we have. Not Nobel Prize work or anything, but the day-by-day experiments that no one else has done. Being able to see the world and be the first person, that's a real gift that we have. I'm going to end this with a quote at the beginning from Abraham Fletcher's article, because that was written in 1939. This stopped really being World War II. And he has a very nice statement about academic freedom. In certain large areas, Germany and Italy especially, efforts are now being made to clamp down the freedom of the human spirit. Universities have been so reorganized that they have become tools of those who believe in a special political, economic, or racial creed. The real enemy of the human race is not the fearless and irresponsible thinker, be he right or wrong. The real enemy is the man who tries to mold the human spirit so that it will not dare to spread its wings, as its wings were once spread in Italy and Germany, as well as in Great Britain.
Thank you, Dr. Chalfie. We have time for 2 to 3, maybe 4 questions. So if you have a question, please raise your hand. I will try to get a student before faculty, though. Yes.
Okay, a couple times already in different groups today, very quickly, it is do something else. If you do more than one thing is sort of at a bottleneck, sometimes the other thing works, and then you can do some enjoyment. We're always thinking of the problems we're working on, never actually abandon them. But sometimes you just have to stop hitting your head against the wall, try something else, until an idea or an approach hits you, as I've said a couple times, when you look in a lab, there may be people that are very happy working in the lab, and there may be people that are unhappy working in the lab. But the head of the lab likes it, at least one person. So, do more than one thing. No one likes that. Oh, we should call.
Yeah, so you mentioned when you were looking at traceless proteins, you heard about the work the other scientists did. So how do you recommend that scientists find out about other people's work, especially like now that there's more and more papers and stuff being published every year?
You know, it's really hard to keep up with everything. You try the best you can. Most of the time, most of the time when I go to places like this and talk to people, I get to hear about what their work is, they get to tell me why it's important. That's something quite different from just reading it in the paper. Right? You get their perspective of why we're deciding about it. So a lot of times, one of the reasons faculty like to go to other places, I like to go to other places, just hear what other people are doing. It's hard. You try journal clubs or journal summary clubs. Sometimes people try to. I've heard of labs. Sometimes people different people in the lab will be responsible for the journals and papers and the attention of the whole group. But yes, it's sort of overwhelming. It's, nature popcorn, this and that, just too many things to keep track of all those the journals and all the try to figure out what it is. But the things like PubMed, you can actually look in the top rather than looking at the journal. But yeah, there's a lot of stuff. A little louder, please. So as a graduate student, I would like to know how do we actually come up with the hypothesis and what should be the driving force or what should be the way to implement your hypothesis? So I'm going to answer that. I'm not sure I'm going to answer exactly the way you want, but I think that often people, as they begin graduate school, often feel, oh, the biggest barrier is I have to come up with my project. And I've never actually found that. When I get ideas, when people in my lab get ideas, it's because we're working on something. So these discoveries that I talk about, they weren't, let's go sit down and have a discovery. It's, I have a goal in mind of this, and oh, that's an interesting result. So for me, work sort of evolves from previous work. And so it's not trying to develop something to know. Every once in a while, however, you might have a really good idea and you want to just start playing with it. But for me, it's sort of a more leapfrog approach.
Are there any resources that are similar in theme to this lecture that we could either read ourselves or point other people to that would kind of evidence how basic science or not
So there's a couple of things. I think that this lecture in one form or another is on YouTube itself. One, how many people know about the Golden Goose Awards? A few. Going to the Golden Goose website might be a nice way of finding out this. The Golden many years ago, there was the Golden Fleece Award, which was a science and something stupid and had to be made fun of. And that sort of went after, at one point, the Golden Fleece Award went to a diabetes treatment that actually helped a lot of people, so they sort of stopped that. But about 2010, two Republicans and two Democrats in the House of Representatives and AAAS, Publishers of Science, and some other organizations, put together the idea that there should be a prize for science that at first sounds somewhat absurd, silly, maybe a waste of time, but it actually turned out to be rather remarkable. Fortunately, I got one for GFP, so. But my favorite is rat massage. With one, and it's really a spectacular story. A group of scientists in the 70s wanted to make a rat strain, and they were very worried about this strain that the mother might kill the pups. So they took the pups away from the mother, and to their horror, they did not thrive. When they gave the pups back to the mother, the pups did quite fine. And so they decided try to figure out why that was. What they realized is that the mothers were constantly licking the babies. So they did an experiment, and they found they did not lick the babies. They found that if they took a Q-tip and just stroked the back of the baby, that giving it some sort of massage, they would still thrive. So the physical touch was important. Why was this given an award? Well, it was realized that this might be a tremendously important thing, and it turned out to be for premature babies all around the world to have that physical contact and to be assisted in this way. So it's a wonderful prize starting off with this really strange result. And I think all of these things, they give about 3 or 4 every year, at least 15, 16 years. So there's a whole series of wonderful examples of this work that didn't maybe make sense at the beginning. One of the other first things they did was give it to Charles Towns for developing the laser. If Charles Towns liked to say that he went to his chair at the Department of Physics at Columbia and told him his idea, and the chair said, don't do that. And he went to the former chair and told him the idea, and the former said, don't do that. But then he did the experiment, and that's eventually what led to lasers. So, not a bad experiment.
We have time for one more question. So, if someone has a question, raise your hand. Yeah.
A lot of students come to me and they have a certain sense of malaise about AI. Right? We just had 200 million structures created, given the Nobel Prize. Plenty of people. And I wonder, I have my own answer, which people don't seem to like. Do you have an answer about why there's going to be a very positive future for science research with AI?
You know, it's like any technology. It's something that can't be just accepted. I mean, I think back at the time when in molecular biology, everyone was using their own enzymes. And all of a sudden, kits came out. People were using kits to do everything. And the big argument was, they don't know what they're doing. They don't know if it goes wrong, they don't know if it goes right. And it's a valid argument. You want to be able to know the basis of things. So I think there's an element there. I told a couple of people, I'm doing an AI experiment in my genetics course, which has turned out to be always said the same thing. The problem with AI is students are going to cheat, which I put as the usual way that basically everybody hates students. They're the ones that are bad. We'll never cheat. Hey, or so I thought, but that's a really silly sort of way of thinking about it. Why not? If you're worried about students cheating, give the essay question to Claude, Chalfie, or the others. Have it write the essay, and then give the essay to the students and say, fact check this. And so that's what I've been doing in my genetics course. We have an internal access to AI chatbots at Columbia, so we're not putting anything on, violating copyright. And we're taking the, I've taken the articles and sometimes figures, and said, write a summary, and I've given it to students and said, fact check this. It turns out they have to read the original paper because they have to find the mistakes. So it's really a terrific learning tool as far as I'm concerned. But it's been very interesting, and they've seen what the hallucinations are. And on the other hand, they're good summaries, but it's being able to understand whether it's a good summary, what's not a good summary, how it's going to be used, where we're going to use this for, what are the limitations. And I think we all need to be trained in to see that, just as we need to be trained in any technology. So that's I mean, anything that allows one to look at vast data sets, I think is a useful tool, because we can't really do that. And so when you have something like AlphaFold, is it perfect? No. Does it need to be improved? Absolutely. But it gives you a push in a particular direction that you decide is correct or not. You can do that hypothesis. So I'm guardedly optimistic and encouraged by things, but I think the blind acceptance is problematic. I have a very good friend who watched her teenage son write an essay for school. Got called in by the teacher, who said to her that she was worried about her students cheating. And so she took all their essays and submitted them to ChatGPT and asked the question, did you write them? Now, it's a large language model. If you ask a yes and no question, about half the time you're going to get yes and half the time you're going to get no. And she said to my friend, and your son cheated. Think about what that means. The teacher goes up to the son, that my friend watched him write the essay and says, you've been cheating. What is his natural answer? No, I didn't. And then she says, now you're lying. And it follows that. And it was all because the teacher believed that she was getting the honest answer from ChatGPT. So I think there are some things to watch out for, be careful about, think about. But on the other hand, it's the opportunity. The machine learning is getting quite a lot. And if we get insights that we can then test experimentally, why not?
Okay, thank you, Dr. Chalfie. You were able to make it out here. We had some hurdles along the way, but it all worked out. And I think it's fair to say that we all realize how useless knowledge might not just be that useless.